So I'm Lisa Vollbrecht. I'm the research manager at Kampachi Farms here in Kona, and I'll be talking about a demonstration project that we're planning to grow native Hawaiian seaweeds, limu, offshore, off the Kona coast here. Specifically, what we're trying to do is culture these limu offshore with no external energy input. So what that means is we're going to culture them with solar, wind, current, wave energies. That is the ultimate goal. And in the demonstration process, we'll show some of the components of those technologies. So we're going to demonstrate at a small scale first, see if it works, see if the technologies work in the open ocean. And if it does work, this is a great way to commercially culture macroalgae. Not only for food, for human food, also for feeds, for livestock, and for fish, and ultimately for biofuels, as that technology allows. So why grow limu? It's a crop that requires no land or fresh water to produce. That's important in areas where those resources are limiting. And it takes up carbon from the environment. And that's very important because dissolved carbon is the cause of ocean acidification. If we grow something that takes up carbon, we can reduce ocean acidification locally. By how much? We don't know. That's something that we'll look at in the demonstration. And of course, we want to grow it because it tastes great and has those applications in feeds and fuels that I've mentioned. And why go offshore? So at Kampachi Farms, part of our mission is to be able to grow the sustainable offshore culture of seafood. We want to be able to move that culture offshore, away from our coastal lifestyles, our coastal resources. If we were to grow a platform of limu, we wouldn't want to do that over a coral reef. It would be like putting an umbrella over it, right? We want to push this offshore, not just demonstration scale offshore, a few nautical miles, but in the large scale, many tens, hundreds of miles offshore even. In the offshore environment, we have ready access to those renewable energy resources. Most specifically, wave action. Consistent wave action is important. It's a constant source of energy. Here in the tropics, we also have solar energy, like there is a few other places on Earth. More beautiful, bright days of sunshine to be able to grow macroalgae. And being offshore means you have access to more of the water column, which is important for this project because here in Hawaii, our surface waters are a nutrient desert. There are hardly any nutrients in them. But as you go deeper in the water column, you start having a higher concentration of nutrients. And we think that the species that are native to Hawaii may opportunistically access those nutrients. So when they're available, they might be able to more rapidly uptake them. So if we could offer them nutrients from deeper seawater for a short portion of the day or once a week, they might be able to grow sufficiently well on that. And this is just a graph showing the relationship between depth, temperature, and nutrients. So as you go down, you'll see that nutrients increase, those are the black dots, and temperature decreases, which are the blue dots coming the other way. So there's an integration there. You only need to go so deep in the water column to access the nutrients you need. You don't need to go all the way to 1,000-meter deep seawater to find that, and we certainly wouldn't be doing that on a demonstration scale. So this is the project concept, and I'll just describe it here. So the mooring system is a tripod, so it's got 3 legs to it, and there's one single point that will reach the surface. The tripod mooring is something that's commonly used in oil rigs, large ocean-going ships. It's a really robust mooring system that uses oil rigs to be able to actually distribute the oil through that swivel point. And here it will be the first time it's demonstrated in aquaculture. And it's of specific interest to us because you can have a 360-degree swivel around that center point. And that means that it's always going to track into the current. It's always going to face its head end into the current, no matter which way it's coming from. So to upwell the nutrients for it, our engineering partners at Mackay Ocean Engineering, I should mention, that are working with us on the design and the deployment of this project, they're also working on a passive wave buoy system that will upwell water. So this buoy here will actually ride on top of the waves as a simple piston pump, the most simple pump you could imagine, just up and down on the waves, and it'll bring a small amount of that deeper water up as it rises. And it'll accumulate it at the head end of the array, and then we can release it, and it will always move down the array because that array is always tracking into the current at a period of time that we develop, once a day, once a week, yet to be determined. And once you grow the algae, you need a way to harvest it. So in the demonstration scale, we're not going to have an autonomous harvester, but we will demonstrate the components of a harvester that's going to be wave and current powered. So sort of like a glorified lawnmower, a Roomba for macroalgae maybe, will move up and down the array as the wind and current allows. And I brought a small video that the engineers did of it tracking with the current. Let's see this. There it goes. So this is a complete 180-degree current shift on the array, just showing the forces at kind of a typical Hawaii day, half a knot of current, a significant wave height, 2 meters, and it will always just clock around. And this is a side view of the project, not to scale, but you can see there'll be the 3 legs of the mooring. We've moved away from integrating the deep seawater pipe with one of the legs because the site that we've selected, I'll show you in just a second, is in a little bit shallower water, so it's going to be a separate leg on the mooring. And this small farm concept will be around 50 meters square, and we're going to deploy it for approximately 3 years. So that's going to give us time to demonstrate this mooring technology, the growth of the algae, to validate that it grows how it does in our test tanks on site here, and to be able to see if these harvesting technologies work in the open ocean. This is the location that we're planning to permit. It's outside of Kailua Bay. It's about 1 nautical mile straight offshore of Old Airport. And you can see in the red circle, there would be 3 points of contact with the bottom in that circle, the tripod mooring. And then there'll also be a small channel for a deep seawater pipe to run further downslope from there. We were specifically looking for a site that was well sheltered from the trade winds and easy access for us. We're here at Nauha. To be coming out of Honokohau and just go around the point and be able to be taking data, visiting it every day is very convenient for us. And what to grow on it after you design all of that. So Dennis and Keely, who introduced themselves earlier, they work on our algae yard down here at Nauha, and they've been culturing a number of different species to decide what might work best offshore. Not everything works well offshore. It needs to be able to handle some wave action, some current. So we've designed some trials to look at what depth these algae grow best at. If it grows best at 5 meters, we can engineer around that and have a 5-meter deep platform. If it likes 10 meters, we can have it a little deeper, right? And also the nutrient requirements. So we want to make sure that these algae can survive in those nutrient-poor conditions with a small blast of nutrients every so often before we go ahead and put it offshore. Some algae need more nutrients than might be supplied in the offshore environment. These are some of the species that we have worked with, some successful, some not so much in the offshore environment. Specifically, Alva here has a cyclical reproductive cycle where it dissociates every 1 to 2 weeks. So you have a strong blade of Alva, and as it reproduces, it basically turns into mush, which isn't the best thing if your crop disappears every 1 to 2 weeks. That's an important fact. And we're also working on sporulation technologies. So there are 2 ways that you can propagate most macroalgae. It could be done vegetatively by fragments. Some species will allow you to do that. The other way is to actually sporulate it, sexual reproduction in the lab. And that's important because it gives you diversity. So on an offshore array, even at the demonstration scale, we'll need many hundreds of meters of line seeded with algae. And you could see the heartburn that might give Dennis and Keely to tie hundreds of meters of vegetative fragments onto the line. So what we're working on is in the lab to be able to mimic those sporulation conditions for the algae, to settle it onto line cartridges. It's basically like a spool of really thin line. And then you can grow them in nursery conditions, get small plants. These are small ulva here on a plate culture. And then take it offshore and you wind that seed line around the structural members on your array. That's how it's typically done at a commercial scale, just not with tropical species. Some of the species that we're working with, nobody has tried these things before. So there's a bit of challenge inherent with that. And then why do it in the first place? So we have species that are delicious foods, especially in poke. These are a handful of the species that we're working with here. For the feeds in both fish food, which I'll talk about, and in livestock, and for biofuels. So to lessen our reliance on fossil fuels as this technology comes along to be able to extract biofuels from macroalgae. And I'll touch briefly on some of the work that we do at Kampachi Farms with an herbivorous fish species. And we're really excited about this because this fish, Nenue, a chub, if you've ever snorkeled in the ocean, you've seen this fish on the rocky shoreline. It only eats limu as an adult. In aquaculture, the carnivores are typically the highly valued, tasty species, but they require diets that are high in proteins and oils. And that's difficult in a nutritionally challenged planet to find sources of protein and oil. Historically, those have come from wild-caught fishery resources, and now research moves along. You can get it from different waste streams, byproducts of aquaculture. There are alternative sources of proteins, single-cell proteins. But the other side of that coin is to be able to grow an herbivore that doesn't have requirements of proteins and oils. So this is an example of a fish that could eat limu directly as a raw food, could eat it in a pelletized format. We're working on ways to develop a diet around that. And it could also be put into a biodigester where you essentially ferment the macroalgae into a single-celled protein. And then it could be fed to animals that have higher requirements for protein. So I want to thank you guys for being here today and listening to this talk. This is part of our public discussion about the project. So I would encourage you, if you have questions, I'd love to hear them. If you don't have one tonight, please take down my contact information. I have some cards. Come and find me. We're asking to do something in the public domain, so we want to hear from all of you what your questions, your suggestions are for the project. Thank you very much.
Your harvester looks a lot like the harvesters you use for kelp.
It does. Okay.
Where are you looking for funding?
So this project is funded through the Department of Energy. It's part of a larger set of projects that they're doing to be able to grow a source of biomass for macroalgae. Because they're working so heavily on the biofuel technology, their concept behind it is kind of like build it and then they'll come, have the biomass and the technology will follow. Right, done.
Can you explain your relationship with Makai a little bit more? I know because what they're doing is complementary to what you're doing, but on a little bit larger scale. Yeah, so Makai they're ocean engineers, so they can design this type of project quite easily. They're also working on the nutrient requirements and the nutrient modeling. So they have the technology to be able to map the shoreline, look at what depth we need to be able to go to access nutrients, to grow them. So it's an equal partnership. They're very involved in this project and the development of it.
Can you go back to the first graph you put on?
Sure. The one with the nutrients?
No, the temperature and nutrient. Yeah,
yes,
that's it.
This one. Yep.
Now the temperature blue, I don't quite follow how that goes. You go from 0 to 40.
It's colder with depth.
This is,
Forward.
yeah, they used the same scale here on the axis, but blue is the temperature. So it goes from
20,
so 20, 24, 25,
Oh, okay,
down to
so
4 degrees.
all right,
Yeah,
okay, so
on
it's
this
going down
axis,
that way.
yeah,
Okay,
yeah.
I got you now. And then you basically, you said there was a pipe bringing up some water. Is that this where you're trying to get the nutrients up there?
That's right, yeah. The pipe that we're designing will probably go to 200, 300 meters depth. So it's very shallow in terms of this graph. We're just looking at this area.
Yeah.
But it's based off of one of the UH stations where they measured much, much deeper. Yeah.
At what point does light become a limiting factor?
To grow the algae at?
Yeah.
Well,
in general.
Yeah, that's one thing that we haven't tested. We haven't looked at a limit to depth, mostly because we want to be able to grow it as fast as we can. So we're not really looking for that lower limit. We're more looking for the upper limit. Yeah, we want it to grow as fast as it can. Yeah.
That's a follow on to that is what depth are you planning to start out at?
Okay, so the species that we're looking at right now that are most promising, the front runner is a gracilaria, and it grows best around 1 to 7 meters in depth is what we're looking at.
And so you plan to maintain the level of your whole system?
Yeah, that would be the idea. Yeah.
These systems aren't really far offshore. The tripod mooring legs look really skinny. I'm wondering if you know what you're going to make them out of to prevent entanglement.
Right. So that is an engineering question, but there will be constant tension on them, and tension is really how you address entanglement. You want a taut line, always. So it will be taut lines with the surface buoy, and that's how you address entanglement. Have big diameter lines, which certainly they will be, so that they don't snap and keep them taut. Yeah. Yeah.
Can you tell us about your background and how you got into this particular management research position?
Oh my goodness. Long story. So my background is in marine and freshwater biology. I worked in conservation for a number of summers in my university career, and I didn't really find that to be addressing the problem enough. Conservation always felt like chasing the tail end of the problem to me, and aquaculture seemed like facing it head on. So I moved to Hawaii and originally seen one of the team members at Kampachi Farms do a presentation on one of our offshore projects, the Villela project, which people in the community would probably remember. It's a big golf ball. It's shaped fish pen offshore. And went up and introduced myself and got involved at Kampachi Farms and began managing the research program from there. This is just one of our efforts. We do a number of other projects on feeds and different species in aquaculture and technologies. Yep.
Will you be teaching tomorrow at the GEMS program for the 5th grade 10-year-old girls?
No, I didn't know that was going on.
Yeah, that'd be perfect.
Camp tomorrow
Okay.
all day. Girls in engineering, math, and science once a year.
Oh, it's only once a year. Oh, I'd love to find out more about it. Do you have a contact that organizes that? Okay, yeah, we should link up back there. Yeah. Thanks.
So we all think that more light is good, but sometimes more light is actually bad when it comes to plants. Photo inhibition is a beautiful thing,
Yeah.
and it's often a complex relationship with nutrient load. And we started getting into that, dissecting that relationship.
We're certainly seeing that integration now. I can't say that we've teased it apart in any large way, but there certainly is too much insulation. They respond to light quite abruptly, and the nutrients are something that we're learning even more about now. Some species are a different color in different nutrient conditions, sometimes the same color that they are when they're stressed in light conditions. So it's, yes, it's an excellent observation, and we are seeing it.
I don't know what you call it, but where you're growing your limu, what are the dimensions?
On our tank trough,
Yeah.
they're 120-gallon tanks. How big are those tanks? Yeah. So they're maybe about 6 feet long, 2 feet wide, 8 feet long. 2 by 8. Go with that. Yeah.
Is your engineering design still in a conceptual stage, or do you have some preliminary ideas about your structures?
So we have some preliminary ideas that we're moving forward with in the permitting process. There are a number of permits that require those drawings, and the engineers are at the final stage of preparing them. We've only just selected this site last week, finalized the site, so they're now engineering around that separate deep sea water pipe.
And are you thinking of a compliant structure or a rigid structure as your substrate?
Oh, you mean the benthos at the site? We think it's all hard bottom. We ran a benthic
No, I
survey.
meant your growth substrate.
Oh, okay. It's going to be lines, and they'll be semi-rigid, probably an HDPE coating or some other kind of rigid coating on a really thick diameter rope. And then we'll hopefully be doing the seeding line where we can just wrap that around the structural members on the array. So you don't actually replace those big heavy lines every time you reseed. It's just that small diameter rope.
And the concept is that the limu will grow upwards and you will harvest that from above, right?
There are a couple of different ways that the harvester could work. We are planning to just harvest the top of it, almost like a shaver mechanism, crossing blades. But you could also be able to harvest around the rope completely. So there are different engineering solutions to that problem. Yeah.
How fast do they actually grow in the water?
Well, that's something that we're looking at. So in our land-based trials, we're able to give them both surface seawater and deep seawater because of this amazing resource that Nelha has here, deep seawater on tap. So we can make our own mix and see how fast they grow. Some of the species we've seen 400% per week in nutrient requirements that we could reasonably supply offshore. They're very fast-growing species. So, yeah.
I'm curious why you chose that site close to Kailua Bay. What other sites did you consider, and why not just slightly north of Honokaha, where you're closer to the harbor, for instance?
Exactly.
So we looked at up near Kauai High Harbor because they have that flat bottom there. Flat bottom is really important for this tripod mooring concept. It can't go on too much of a slope, and that is part of the challenge. We also looked at a site that was just tucked under Keahole Point, but the bathymetry there is so steep you could never put anything. We did not look at putting something right outside of the harbor. We looked at potentially the conflict with boating and recreational boating.
Yeah, there's a lot more boating traffic there.
Yeah, so we're looking down in Kailua Bay.
What's the depth where you're going to be?
Yeah.
The center point of that site that I showed is about 120 meters. Just pull it back up again. And this chart has soundings in fathoms. So any number on the chart, you multiply it by 6 and you could get the depth in feet. The center point's around 120, and then the deep sea water mooring would go to about 200-300 meters.
About how far offshore is that?
So this edge of the circle here is about 1 nautical mile straight offshore. But the growth platform itself is going to be about 50 meters long, so the surface structure isn't going to be quite that close to shore.
You don't worry about the cruise ship going by there?
So this is actually the cruise ship channel right here. So
okay.
This is the channel they moved down, and they've got both a green and a red zone that they have to course correct in. Okay. So yeah, they come in that way. It does seem like they come right around the corner, but this is actually
really that far.
You mentioned biofuels. Do you expect to grow lipid-rich algae and extract the lipids, or are you just going to put the limu up and burn it, or what's your ideas about that?
So hydrothermal liquefaction is the most commonly used way to make biofuel out of macroalgae. That is not our area of expertise, so I can't talk in great detail about that. This project will be doing some bomb calorimetry just to see bulk levels of energy, what energy comes out of it, so we can look at the energy returns, the renewables that we're using to grow it versus how much energy we're getting out of it. But in terms of actually developing the biofuel process, that's not necessarily under the project. Yeah.
Somebody else's career.
Given that this is so much focused on energy efficiency, it seems to me that you might be missing out on using kinetic energy from the currents that you're going to be sitting in. Do you know if Mackay are looking at maybe harvesting a little bit of that kinetic energy as in addition to the wave?
So the way that we discussed using current energy is primarily in harvesting to be able to essentially cut it at one end and collect it at the other end. But you're talking about generating electricity from the current.
Using that drag to generate, probably pumping up, because I'm thinking that basically the more deep water you can pump, the faster you can make it grow, the more efficient you're growing. And so any way that you can get more deep water up is good.
Yeah, I'm not familiar with many of the current kinetic technologies. You'll have to school me on that. Go ahead, Doug.
Actually, there was originally, you'll remember, the Mackay folk had that piston-powered pump that was going to be current-driven.
Oh, you're right.
That was
the
early conception.
Okay, yeah.
And they thought, no, they really want to go with the wave pump. So that they have looked at it. And
I think that there was some energy demand on the current technology that was priming it or something. It needed some kind of energy input. I can't quite put my finger on it.
yeah.
Candy, go ahead.
Do you foresee any problems with wildlife eating all the algae? Are you gonna do any tasing or any like you show up in the morning and like there's 100 sea turtles
Restaurant.
Yeah, it's
quite
drive
possible,
through.
and we don't know until we demonstrate it.
Okay.
I will call all the sea turtles if I see them.
I'm curious why Kampachi's doing this.
So you're right to ask the question. It's not Kampachi. It's not necessarily critical, but really, on the larger sense of the mission of what we do, is to try and find ways to address what's going on in the environment. And this is a real way that we can apply ourselves and our expertise around biology and around offshore aquaculture to be able to tackle some of those problems, to be able to produce food here. We import seafood to Hawaii. How crazy is that? To be able to tackle some of that problem and moving forward.
Yeah. Okay.
So,
How deep do you think you have to go with your intake pipe to get any nutrient enrichment? I mean, it looked like from your graph, you really have to go down a couple hundred meters in order to get enriched water.
right. Well, it's not an easy question because it really depends on how rapidly the limu can uptake the algae, how deep you need to go. If it requires a whole bunch of nutrients, then maybe you would need to go deeper. But we think that it doesn't require as much nutrients. So we're testing it at lower levels, and we're planning to be able to do this at about 200 or 300 meters of upwelling, not actual deep seawater like we have here at Nalha comes from 600 meters.
So you don't anticipate that your growth is going to be nutrient inhibited?
No, it certainly will be nutrient inhibited, but there's an integration for cost. If you want to go all the way to the bottom of the ocean, it's going to cost you a whole lot of money. So you only want to go as deep as you need to be able to grow the algae fast enough to make it an economically viable project. Yeah.
So for this size pilot project, what size pipe was Mackay looking at? I'm just curious.
For the deep seawater pipe. So they've modeled for a 6-inch pipe and for an 8-inch pipe, so it'll be somewhere in that range. Yeah.
So just that the first pipe that was ever used for deep sea water was 6.
Really?
3-inch.
Well,
in the Virgin Islands.
yeah, in the Virgin Islands, they did 3 and 6 inch. That's getting back to the basics here.
Yeah.
Where are you at in the permitting?
Where are we at? So we've engaged with a number of the agencies, both state and federal, and now that we've selected our site, that's fairly recent, we're going to start actually making those applications in the coming weeks. So we've engaged, we're probably optimistically about 6 months away from anything potentially happening. There will be public hearings associated with it, will be in the community more.
Just saying, people in this room can help with that process.
Yeah.
Is dulse a possible species that they grow the abalone on?
So dulse grows in really cold water, and it's also a non-native. So we're only looking at growing native species offshore. Certainly that's a delicious species though. Have you eaten it before?
Did I understand you correctly when you said that the technology for seeding the array was something that someone else has done before?
Yeah, it's done in other types of culture, cold water culture, commonly. It's just that for the tropical species, very little is known. I mean, when we started researching this project to be able to apply, we looked and did all the research we could on Hawaiian limu, thinking there's got to be a textbook about this somewhere. There isn't. Very little is known. So, we're doing the experimentation, and maybe Dennis and Keeley will write the textbook.
Have you approached the military for funding?
No.
Because they're big on biofuel, Navy, the Air Force.
Are they? Yeah, but that could be something in the future.
The grant that you have now, is there follow-on opportunities for additional years?
So it will have one follow-on, which would be the deployment phase, but after that, they expect you to have something to commercialize, right?
It's like an SBI.
Yeah.
I lost it.
I'll be around later.
Yeah. Oh, I know it. So how much do you anticipate? So if you're bringing up nutrients from below, how much do you think will be absorbed by the limu that you want, and how much will then flow out in the current to wherever else and be nutrients for something else?
So that's a really complex modeling question, and that's something that Mackay, our partners, they're actually doing a separate project on nutrient modeling to see what the requirements are and how much you need to reasonably provide for them to uptake. How much could they reasonably use in a set period of time, right? Because you don't want to upwell more than that. And so I don't have a hard and fast answer to that question, but we're looking at it.
And what happens to all those nutrients that do flow away from the site?
Well, the idea is there wouldn't be any. You only want to upwell and use energy and resources for those that are going to be used by the algae, uptaken by the algae.
Thank you.
Thank you so much.
Great, thank you.
Yeah.